Hi everyone, and welcome to the BME2104 lecture on DNA replication. We've already covered the basics about DNA in our cell parts list lecture, but today we're going to go into lots more detail about the structure and properties of deoxyribonucleic acid, DNA. We'll talk more about chromosomes and chromatin, and then we'll start our discussion of DNA replication by going over what happens at the origin of replication. This is our first in a pair of lectures. We'll cover the rest of DNA replication on Thursday. In the larger scheme of things, this is the point in our course where we begin our series of lectures on the central dogma, which of course goes from DNA to RNA to protein. The human disease topic I'm going to use today is HIV/AIDS, which fits into our theme of focusing on viruses over the semester, but the reason I include this today is because the HIV virus illustrates a result that surprised a lot of people when it was first discovered, and that's that the central dogma can be reversed. So let's introduce the disease AIDS, which is short for Acquired Immunodeficiency Syndrome. AIDS is caused by the HIV virus that you can see on the right here. Like the SARS-CoV-2 coronavirus, HIV is an RNA-based virus that also has a lipid bilayer envelope. Its mechanism of replication is quite different from SARS-CoV-2, however. This involves how it reverses the central dogma, which we'll talk about at the end of class. If untreated, the fatality rate for AIDS approaches 100%, but thanks to years of basic biomedical research, we now have effective drug treatments that have turned HIV infection from a death sentence into a chronic but manageable disease. That said, HIV/AIDS is still an enormous problem worldwide, with approximately 38 million people suffering from the disease. We have 1.2 million people infected in the United States, and as you can see from this map on the right, Sub-Saharan Africa is currently the biggest hotspot for HIV infection. Let's talk now about how the HIV virus spreads. We already talked about how SARS-CoV-2 spreads through contact transmission, and how we can prevent this by washing with soap and water. Remember that airborne transmission for SARS-CoV-2 is especially important as well, which is why we also need to wear masks in public and avoid proximity to other people outside our home and family units as much as possible. HIV transmission is different. It's present in bodily fluids like blood, semen, and breast milk, so it's primarily transmitted by more intimate contact, through unprotected sex or through sharing needles among drug users. For this reason, there was a lot of stigma around the disease at the beginning of the epidemic in the 1980s, but this also sparked a lot of advocacy and activism to fight the disease, especially among the gay community, which was hit very hard at the beginning of the epidemic. HIV can also be transmitted by breastfeeding and blood transfusions, although this last one is more historical because very early on in the AIDS epidemic, we started testing all donated blood for the virus. Symptoms of HIV infection include flu-like illness, including fever, muscle aches, sore throat, and mouth ulcers. Progression from HIV infection to AIDS disease is characterized by more fever, swollen lymph glands, weakness, weight loss, and an increasing number of severe illnesses from opportunistic infections, which is due to the immune system being weakened. Remember that AIDS stands for Acquired Immunodeficiency Syndrome because it attacks and weakens our immune system. I'll go over this more on the next slide. One of the opportunistic infections that was frequently seen with AIDS before modern treatments were developed is Kaposi's sarcoma virus. This is a virus that causes cancer, similar to HPV that we'll talk about later in the semester during our lecture on cancer and cell signaling. This is just one example of all the opportunistic infections that can come in and eventually lead to fatality when the immune system is weakened. So let's talk about how HIV weakens the immune system. Similar to SARS-CoV-2, it has specialized proteins in its viral envelope that recognize and bind to cell surface receptors. This facilitates virus internalization into the host cell. One of the cell types in our body that gets infected is CD4 T cells, also known as helper T cells. We'll cover these in much more detail during our lectures on immunology later in the semester. But for now, all you need to know is that these are specialized immune cells whose job is to help other immune cells fight infections. That's why they're called helper T cells. We've actually already seen these before in class. They're an example of leukocytes, which are really small compared to neurons. Remember I showed this size and morphology comparison between those two cell types on the first day of class. Alright, so progression of AIDS disease is characterized by depletion of CD4 T cells, which leads to the weakened immune response that's characteristic of the disease. And how the HIV virus progresses through its life cycle inside these cells is something that we'll come back to at the end of class. Instead of just replicating its RNA genome, HIV actually converts its RNA into DNA and then inserts this into our genome, effectively reversing the central dogma. We'll go into the details of how this works at the end of lecture today. Alright, so let's begin our sequence of lectures on DNA replication and the central dogma by going into more detail about the structure and properties of DNA. DNA is of course made up of nucleotides, and all nucleotides have a common structure with three components: an aromatic base, a ribose or deoxyribose sugar, and then one or more phosphate groups. Let's go over these one at a time. There are different aromatic bases, but they all look a little bit like the adenine molecule that's pictured here. Aromatic bases are aromatic. That is, they have this nice, cyclic, conjugated electron system that you can see here. Aromatic bases are also bases. They have a basic nitrogen that is used to form a covalent bond to the sugar of the nucleotide. In this picture, the basic nitrogen of adenine is at the 9 position. When we think about nucleotides, we always have this linkage of the aromatic group to the sugar group at this basic nitrogen atom. So in nucleotides, we don't actually see this acting as a base, it's just part of the bond instead. Okay, so now let's move on and talk about the sugar group. This will be ribose in RNA and deoxyribose in DNA. An easy way to remember this is just from the full names of RNA and DNA. That's ribonucleic acid and deoxyribonucleic acid. So as you can see here, ribose is a 5-carbon sugar that forms this nice cyclic ring structure when the 4' hydroxyl attacks and bonds to the 1 carbon. That becomes the oxygen atom you see at the top in all of these drawings. The difference between ribose and deoxyribose is illustrated here. Deoxyribose is just missing a hydroxyl or OH group. That's why we call it deoxy. In DNA, the hydroxyl group is missing from the 2 position of the sugar ring, so this is called 2-deoxyribose. Let's talk for a second about this numbering system. The numbering of atoms in adenine and the sugar rings comes from rules used in organic chemistry that we're not going to cover in this class, but I do want you to be familiar with the numbering on the ribose sugar ring structure. These numbers are important because they'll define how we talk about the directionality in nucleic acid polymers, just like we use the amine and carboxy groups of amino acids to talk about the N-terminal and C-terminal directionality in proteins. So here we have the 1, 2, 3, 4, and 5 position carbon atoms. I want to bring your attention to the 5 position because this is the position of the phosphate group on a nucleotide. All nucleotides will have one or more phosphate groups linked here. As we'll see in a couple slides, it's this 5 position and the 3 position that become important for the directionality of nucleic acid polymers. In the picture here, we have adenosine monophosphate with only one phosphate group. That's also known as AMP. From our lecture last week on enzymes, we've already talked about ATP, which is adenosine triphosphate. Remember, this is the phosphodonor for kinase enzymes. ATP looks like the AMP molecule pictured here, but it has 3 phosphate groups in a row instead of just one, like this. As we'll talk about later in the semester during our lecture on metabolism, ATP is also the primary currency for energy in the cell, so it's really a triple threat in terms of important jobs. It serves as an RNA building block, it's a kinase phosphodonor, and it's also the molecule used for almost all the energy-dependent processes inside the cell. When we're talking about these molecules, it's important to be precise in how we describe them, so let's go into a little more detail here. If you take away the phosphate groups from a nucleotide, what you're left with is a nucleoside. This is just the nitrogen-containing aromatic base and the ribose or deoxyribose sugar. One thing that you'll see as a naming convention is that nucleotides are often referred to as nucleoside phosphates. We've already done that with adenosine monophosphate and adenosine triphosphate on the previous slide. Adenosine is the name of the nucleoside. Okay, so now let's talk about the different bases that we'll see in nucleic acids. There are 5 of these that you'll need to know about, 4 that make up the genetic code of DNA, plus 1 more that's used in RNA. These bases come in 2 major categories, purines and pyrimidines. Purines on the left have this double ring structure with a 5-membered ring and a 6-membered ring linked together. the two purine bases are adenine and guanine. The other major category of nucleic acid bases are pyrimidines. These are single six-membered rings, and these include thymine, cytosine, and uracil. You can see the one-letter abbreviations for each of these bases given here. You'll see these used quite frequently, and unlike amino acids, it's simple to remember them because they're always just the letter of the base that they start with. So make sure you learn these one-letter abbreviations. That's something we expect you to learn for this class. A quick note about the structure of these bases. If you look closely, you'll see that the three different pyrimidines are identical at the numbered positions, but vary in their outer constituents. The same goes for the purines as well. All these bases have carbons and nitrogens as part of their ring structure. And one more thing to note is that they all have a basic nitrogen, colored in pink here, that forms the attachment to the ribose or deoxyribose sugar. Okay, so now let's talk about which of these bases are in DNA and which are in RNA. In DNA, we'll see the bases adenine, thymidine, guanine, and cytosine. Just those four bases make up the alphabet in which our entire genome is, all the genetic information in our genome is stored. RNA molecules are the same, except they use uracil instead of thymine. So thymine you'll only see as a base in DNA, and uracil you'll only see as a base in RNA. Okay, and one last thing on this slide that I'm giving you is the table here with how we name these bases depending on whether it's a base alone or coupled to a ribose sugar, in which case we add this O-S-I-N-E or D-I-N-E sound to the end. So adenine becomes adenosine or deoxyadenosine. Cytosine becomes cytidine or deoxycytidine, etc., so on and so forth. You don't need to memorize all of these names, but I want you to be able to recognize them as they come up in your reading or in future class lectures. Okay, so we've talked about nucleotide monomers. What I'm going to tell you about now is how these monomers get linked together to form long, linear polymer chains. The bond that holds these nucleotide monomers together is called a phosphodiester bond. This is the covalent linkage between the phosphate group on the 5' carbon of one sugar and the hydroxyl group on the 3' carbon of another sugar, as you can see here on the right. To put the phosphodiester bond into context some of you may have seen before in organic chemistry, I'm showing here on the left a structure of what I'd call a normal ester bond. That's the carboxylate ester. Next, we have a diester bond, which has a double bond carbonyl with oxygens on either side of it. And finally, we come to phosphodiesters. This is a diester bond that substitutes a phosphate group in place of the carbonyl. Okay, so in DNA and RNA, this type of phosphodiester bond connects the 3' position of one nucleotide with the 5' position of another nucleotide. Going along these lines, this linkage can be made over and over again to give rise to long, linear nucleic acid polymers. Because this 5' to 3' linkage is asymmetric, this gives the entire nucleic acid polymer a polarity, just like protein polypeptide chains have an N-terminal and C-terminal polarity. It's the same idea here. So when we read out a nucleic acid sequence, we always read it out from the 5' end to the 3' end. That's the convention that we use, just like we always read out protein sequences from the N-terminus to the C-terminus. This choice of direction isn't arbitrary. It's because DNA molecules are always synthesized in our body. They're always polymerized in the 5' to 3' direction. That's why we read out the bases in this direction as well. One last thing to mention on this slide. Looking at the picture on the right, you can see the repeating phosphate and sugar groups. This is sometimes referred to as the phosphodiester backbone. Just like proteins have a fixed peptide backbone with varying amino acid side chains, DNA and RNA will have fixed phosphodiester backbone with varying base groups, as pictured here with C, A, and G. You should be able to tell from this picture that we're looking at a DNA molecule, not an RNA molecule, because the ribose sugars don't have hydroxyl groups at the 2 position. So this is a polymer of deoxyribonucleotides. One of the most important discoveries in the history of molecular biology was the structure of DNA polymers in the cell. This double helix that you can see on the left here, consisting of 2 DNA polymer strands that wrap around each other as they twist up into this double helical shape. This was so important because the structure helped us figure out exactly how genetic information is stored in the cell. It also helped us figure out how it's replicated during cell division and how it's translated into RNA for protein synthesis. We're still very much living and the molecular biology revolution that was sparked by this discovery, cracking the genetic code, sequencing DNA, predicting protein sequences. A very large portion of what I teach you in this class would not have been possible to learn and understand without this very basic and fundamental piece of information about DNA structure. This was first published by James Watson and Francis Crick in 1953, who famously didn't give enough credit to Rosalind Franklin, even though she was the one who generated all the X-ray crystallography data that led to the model and shared her unpublished results with them. So that's a little bit of the history. Let's talk about this structure now. It's a right-handed helix with the phosphodiester backbone pointing out and the bases pointing in. Thinking back to our lecture on protein structure for a second, this is the opposite to alpha helices, which have their side chains facing out and the peptide backbone on the inside. In the stick figure diagram on the left, yellow is for the phosphorus atoms in those phosphate groups, and blue is for the nitrogen atoms in the aromatic bases that are stacked in the middle. Watson, Crick, and Franklin didn't discover this full atomic scale structure, but they did come up with the very general model of the double helix structure with bases in the center. And this gave them the idea that the bases paired together by hydrogen bonds that are specific to their geometry. So guanine and cytosine only pair with each other, and thymine and adenine only pair with each other. These are called base pairs. This is a blue term that's important for you to know and understand. This insight about base pairs was really the key to unlocking the genetic code and understanding the central dogma. So let's talk about all of this a little bit more. As you can see in these pictures, each base pair consists of one purine and one pyrimidine. This is important because two purines would be too large to fit in the center of the helix, and two pyrimidines wouldn't be long enough to bridge the gap, but the purine and the pyrimidine together makes it a perfect fit. Another thing I want you to notice is that the molecular force holding the base pairs together is hydrogen bonds. GC base pairs have 3 hydrogen bond interactions, but AT base pairs only have 2. As a consequence of this, the GC base pair is a stronger interaction, and DNA sequences that are GC rich will be harder to pull apart. We'll talk about this process of pulling apart DNA strands later in the lecture. As you can see in the structure on the left, is the base pairing hydrogen bonds that hold the DNA double helix together. Individually, any one H bond isn't so strong, but when you add them all up, the stabilizing force they provide for the double helix structure is considerable. One last thing to mention on this slide is that we use the number of base pairs in a DNA double helix to measure how long it is. The structure on the left would be around 13 base pairs long, although this is a little unclear because near the bottom only one strand is displayed. Okay, so just to reiterate, it's hydrogen bonding interactions that provide the force holding the DNA double helix together. When you have two different strands here with bases matched up in base pairs, you can see the matching of the A's to T's and the G's to C's. These two strands are complementary because they form a perfect match of base pairs. One thing that's very important to notice here is that these two strands are anti-parallel. Remember how we discussed that the nucleic acid polymers are directional because of their 5' to 3' phosphodiester linkage? Well, if you look at these two nucleic acid polymers, you'll see that they're running in opposite directions. The one on the left goes up in the 5' to 3' direction, and the one on the right goes down in the 5' to 3' direction. We refer to this reverse orientation in the double helix as anti-parallel. I can take this molecular structure and convert it into a schematic diagram for clarity. If you have one DNA strand that has the sequence ACTG, its complement strand will have the sequence CAGT, remembering that the convention is to always read out DNA sequences in the 5' to 3' direction. So again, DNA exists as complementary anti-parallel strands like this, not parallel strands like this. A parallel strand DNA double helix is not structurally possible. Okay, so here are some more diagrams of anti-parallel DNA double helices. The point I want to make here is that the hydrogen bonds that hold the complementary strands together can be broken apart. They're electrostatic in nature, and you can unwind the two DNA strands just by heating them up. We do this in my lab all the time. We take DNA and bring it to 100 degrees Celsius, which is the temperature of boiling water. When we do this, the individual nucleic acid polymers are fine. They don't degrade or fall apart, but they do separate from the double helix native state into single strands. This process is called denaturation, and the single strands are in a denatured state. This is different from protein denaturation. That's when a protein loses its structure and becomes non-functional in a usually irreversible way. What's happening with DNA denaturation is that the two strands are coming apart, but if we bring our sample back down to room temperature, the complementary DNA polymers, the complementary DNA strands, will rebind to each other in the same complementary anti-parallel orientation. This is called annealing. In my opinion, it's crazy to think about two randomly oriented strands coming back together and finding exactly the right complementary orientation to reform the double helix, because the entropic cost for this is so high, there's so many degrees of freedom. What's probably happening here is there's a lot of what some people call zippering behavior going on. Local regions of complementary sequence bind to each other and then zip up further along as all the base pairs match. Okay, so that's denaturation and annealing. This ability of DNA to denature and anneal back and forth like this forms the basis for an extremely powerful laboratory technique called polymerase chain reaction, or PCR. We can use this for DNA amplification, DNA sequencing, and the construction of new artificial DNA sequences. We'll cover this in more detail during our lecture on molecular methods, and you'll see that this forms the basis for the COVID test that you all took before coming back to grounds, so stay tuned for that. Okay, so that's what DNA looks like and how it behaves at the atomic level. Now we're going to move up in scale and talk about a higher level of organization for DNA in our cells, and that's the chromosome. In eukaryotic cells, including our own, chromosomes are long, linear, double-stranded DNA molecules where all of our genetic information is stored. How long are these chromosomes? Well, the longest human chromosome is approximately 249 million base pairs in length. Prokaryotes, like bacteria, are a bit different. Instead of having linear chromosomes, they have a single circular chromosome that carries all of their genetic information. We humans have 23 different pairs of chromosomes, which is 46 total. Each pair includes one chromosome inherited from your maternal side and one chromosome inherited from your paternal side. Because of this pairing, this is what makes us diploid organisms. We have two copies of each chromosome. Prokaryotes, like bacteria, are haploid, meaning they only have one copy of each chromosome. Very generally speaking, being diploid is a bit wasteful for cell resources because you have to maintain two copies of everything. But we gain a big evolutionary advantage from population-level variability due to genomic shuffling. Being haploid, on the other hand, is very bare-bones inefficient, which is good for organisms like bacteria that need to minimize their energy expenditure and resource consumption because they're often in an environment of limiting resources. Okay, so a bit more about diploid versus haploid. On the right here, I have a picture of Xenopus frogs. This is a diploid organism just like us, shown here at a very early embryonic stage of development. If we use a laboratory trick to force the frogs to be haploid, you can see that they still develop to some extent. They have every gene they need, of course, but diploid organisms are used to having both copies of every chromosome. So if they're missing one copy, this leads to problems and developmental defects. On the right, this picture shows the stage at which haploid Xenopus embryos go into developmental arrest. We'll talk later in the semester about how cells in our body control whether a gene is expressed from just one chromosome or both chromosomes, and what mechanisms control this. That'll be in the upcoming lecture on epigenetic regulation that I refer to as central dogma elaborations. Okay, so our chromosomes are all stored in the nucleus of our cells, but there's another place we have DNA as well. That's in the mitochondria. Our mitochondrial DNA is a single circular piece of DNA approximately 16,000 base pairs long. Our mitochondrial DNA encodes around 37 genes that we use for a few different things, including components of the electron transport chain that we use for oxidative phosphorylation, and specialized versions of the machinery used to translate mitochondrial RNA into protein. We'll cover translation and oxidative phosphorylation in upcoming lectures later in the semester. An interesting thing about mitochondria is that we have very strong circumstantial evidence that the original mitochondria was an endocytosed and internalized prokaryotic organism. Rather than degrading it in the lysosome, we somehow evolved a symbiotic relationship with it. You can see this in its circular DNA. It has sequence similarity to other prokaryotic organisms. Of course, Over millions of years of evolution, most of the original prokaryote's genome has been lost, because most genes were no longer needed. Presumably, the remaining 37 or so genes provide some kind of selective advantage to the eukaryotic cell. Another thing that's interesting about the mitochondrial DNA is that it's almost exclusively inherited from your maternal side. This fact can be used for maternal lineage testing and also explains why there are mitochondrial genetic disorders that are inherited exclusively on the mother's side. Regarding these mitochondrial genetic disorders, some of you may have seen or heard about a new technique in the past couple of years called mitochondrial transfer, which results in so-called three-parent babies and is used to correct mitochondrial defects. This technique has been used successfully in the UK, but is currently not approved as treatment in the United States. Okay, so why do we inherit our mitochondrial DNA from our mother and not our father? We can answer that question by looking at the structure of the male gamete, the sperm. This is much smaller than the maternal egg, and it has very little cytoplasm. So already we're talking about a huge dilution of paternal mitochondria after fertilization, which is of course the fusion of the male and female gametes. But on top of that, there are additional mechanisms by which paternal mitochondria are actively removed. These appear to vary from one organism to the next, but can include degradation of mitochondrial DNA in the sperm, prevention of sperm mitochondria from entering the egg in the first place. The mitochondria you see here, those are in the mid-piece. You can think of that as the mitochondrial engine of the sperm, but this doesn't often enter into the egg after fertilization. On top of these mechanisms in the sperm gamete, there are also active mechanisms in the fertilized egg, including selective degradation of paternal mitochondrial DNA and degradation of the entire mitochondria. Alright, so we've talked about chromosomes and mitochondrial DNA. What I'm going to tell you about now is how we fit these long double-stranded DNA molecules into the nucleus, or into the whole cell in the case of prokaryotes that don't have a nucleus. For example, if we take an E. coli chromosome and stretch it out, it has a linear length, or contour length, of around 1.3 millimeters. The diameter of a bacterial cell, though, is just a few microns, so the E. coli DNA is approximately 1,000-fold too long to fit into the cell. So how does it all fit? We need to have some kind of efficient packaging and compression system. An additional consideration is that we need to make sure it doesn't get tangled up too badly. That'll become very important when we get to DNA replication. We'll talk about the mechanisms for this compression and compaction over the next couple slides, but first, I want to ask all of you to calculate the length of DNA in the human genome. We have about 3 billion base pairs in our genome, and if you look back to the slide with the structure of the DNA double helix, you'll see that the distance between each base pair and the linear double helix strand is approximately 3.4 angstroms. An angstrom is 10 to the negative 10th meters long, and it's a useful unit of length when we talk about molecular distances because it's around the same length as a carbon-carbon bond. If you're talking about distances like this all day, it's just easier to say 3.4 angstroms instead of 0.34 nanometers. Okay, so with the information I've just given you, I'd like you to quickly calculate the length of a single stretched out human genome. You can pause the video to work this out. It shouldn't take too long. All right, welcome back. So by my calculations, we have 3 times 10 to the 9th base pairs multiplied by 3.4 times 10 to the negative 10 meters, and this gives us almost exactly 1 meter. So that's around how long our entire genome is, 1 meter. Of course, because we're diploid organisms, we're going to have 2 meters of DNA packed into every nucleus in our body. Remember that these are around 5 microns in diameter, so this is about a 400,000-fold level of compression, which is quite amazing when you think about it. As a quick aside, another thing to think about is the total length of DNA in your entire body. In order to calculate this, we need to know the total number of cells in the human body. It's hard to get an accurate measure of this, but most estimates are somewhere around 40 trillion cells in an adult human body. So if we think about 2 meters per cell, this will translate into around 80 billion kilometers, which is 10 times larger than the diameter of the solar system, which is also pretty crazy if you ask me. Okay, so how do we take a polymer that's 2 meters long, which is taller than most of us, and compress this down into a nucleus that's only 5 microns in diameter. That's what we're going to talk about now. But before we get to linear eukaryotic chromosomes, let's talk for a second about circular DNA, because this has a special property called supercoiling. How this works is kind of like how an old-fashioned telephone cord gets twisted up. I bet that most of you have never seen this phenomenon before because no one has this kind of phone anymore. But I have one of these telephone cords in my office, and I made a video about this last year. So let's take a quick intermission from these slides to check out my video demonstration on supercoiling. Hi everybody, and welcome to a video supplement for the Central Dogma pre-lecture. What I'm going to show you today is a quick demonstration of DNA supercoiling. I've taken my telephone cord from my telephone. I had to remove all the wires in order to do this. This is from my office telephone. And you can see this is a reasonably good model for DNA. It's helical, which is nice. And what I'm going to do quickly is I'm going to hang it like this. I'm going to let it become completely relaxed. And if we take this now and circularize it, you can see that we have a nice relaxed circular piece of DNA. Now to demonstrate supercoiling, what I'm going to do is I'm going to start twisting it. In my experiments earlier, I've discovered that around 12 times twisting is good to get supercoiling. So let's do this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. All right, so then this is pretty twisted up. There's a good amount of tension on this. You can see. So now we have a linear DNA molecule. I'm going to make it circular, and we'll observe the supercoiling. All right, there you go. That's a video demonstration of circular DNA supercoiling. Thanks for watching, and I will see you guys in class. All right, so I hope that was a clear demonstration of how introducing twists into a circular chromosome will cause it to bundle up and fold on itself. This phenomenon is called supercoiling. In lecture on Thursday, we'll go over some examples of how these twists can be introduced during DNA replication. One other thing to consider is whether the supercoiling is positive or negative. Positive supercoiling means that the twists are in the same direction as the DNA helical twist, and this will lead to even more folded up structure that compresses the DNA even more and reduces the possibility for normal DNA-protein interactions. Supercoiling is a good way to compress DNA, but there's a problem that we need to work against here, and that's the negative charge on the phosphate groups in the phosphodiester backbone. Compressing the DNA brings all of these negative charges into close proximity with each other, which results in a lot of electrostatic repulsive force pushing back against the compression of the DNA. So this is a problem. How do cells solve it? Well, prokaryotes with circular genomes have positively charged DNA binding proteins that neutralize the negative charge on DNA and facilitate higher levels of compaction. We also have positively charged DNA binding proteins like this in our body. In eukaryotes, these proteins are called histones and serve a similar role to help our long DNA molecules become more compact by offsetting their negative charge. Let's talk a little bit more about these histone proteins because they're so important for the organization and structure of eukaryotic genomes. As I mentioned, they're positively charged with lots of lysine and arginine residues on their surface. Histones self-assemble into an octamer of quaternary structure, each one with 8 subunits. There's different types of histone proteins that go into this. As you can see here, there's 4 different main subunits that each appear twice in the octamer. Don't worry about memorizing their names, but just for completeness, these are histones H2A, H2B, H3, and H4. The histone octamer serves as a positively charged surface that DNA wraps around, approximately 1.7 times. This combination of histone octamer and DNA is called the nucleosome. In each nucleosome, about 150 base pairs are wrapped around the histone, and if you include the adjacent sequence of linker DNA, each nucleosome occupies around 200 base pairs of DNA in the genome. Histones are very interesting and complicated proteins. In addition to helping with DNA compaction, they also play a very important role in the regulation of DNA accessibility. We'll talk about this more during our lecture on epigenetics later in the course. DNA accessibility is very important because this controls which genes can be expressed, that is, transcribed into RNA and then translated into protein. That's what I mean when I say expressed. If the machinery for RNA transcription in our nucleus can't access part of the DNA molecule because it's too compacted, then there's no transcription of genes from that region. Like I said, we'll talk about this a lot more later in the semester, but for now, let's just say that controlling the level of DNA compaction, which includes wrapping up or removing histones, this is an active, regulated process that has a big effect on what genes are expressed in every cell, and therefore plays a huge role in controlling cell identity and function. Okay, I just introduced histones as an important DNA binding protein, so what we're going to get into now is a discussion of chromatin, that is DNA and all of its associated proteins. Histones are some of the most important proteins here, but there are many others as well. Some provide more compaction and structure, others provide enzymatic activity or aid in the mechanics of DNA transcription and replication. One thing I want to tell you about on this slide is that there are different classifications of chromatin depending on how compact it is. In the nuclei of our cells, these different types of chromatin are often spatially organized together. So let's talk about this using the electron microscopy image of a nucleus on the right. As a reminder, the darkest region of the nucleus in this picture is the nucleolus, where ribosomes are synthesized and assembled. We already went over the nucleolus during our cell parts list lecture. This is kind of a special case in the nucleus that looks very electron dense because all of the ribosomes are in close proximity to each other. But if we look at other regions of the nucleus besides the nucleolus, we can divide these roughly into two different classes of chromatin. The first one I'll tell you about is euchromatin. This corresponds to the light regions in the electron micrograph. There are nucleosomes in there, but the DNA in this region is not further compressed than that. Euchromatin generally corresponds with regions of active DNA transcription because the DNA is more open and accessible. That means that RNA transcription machinery can get in and do its thing. On the other hand, we have these darker, more electron-dense regions of the nucleus. These are referred to as heterochromatin. What's happening here is that the genome has been further compacted to a point where it's not accessible to the transcriptional machinery, so the genes from these regions will be transcriptionally silent and inactive. What I showed you on the previous page was the nucleus of a cell in interphase of the cell cycle, that is, not actively dividing. When a cell gets to mitosis and it's time to actually divide, our chromosomes condense and compress even further into these structures called chromatids. This is the highest level of compaction that we know about for DNA. You can see the levels of organization in this diagram. We have the double helix, then nucleosomes, then a folded up 30 nanometer nucleosome fiber, which further loops and coils into the dense, highly compacted chromatid structure that you see here, with an approximately 15,000-fold compression ratio compared to interphase DNA. These chromatids are compact enough now that they can all be lined up at the center of a cell and then pulled to opposite sides during cell division. We'll talk about this a lot more later in the semester during our lecture on cell cycle and cancer. So that's all for now about the structure and properties of nucleic acids. I want to come back now to the central dogma for a second, because we're about to talk about something that's related to it, but not part of it, and that's DNA replication. Both DNA replication and RNA transcription involve nucleic acid synthesis. As we'll discuss on the next few slides, new DNA and RNA chains are produced by copying from a template DNA strand. We have specialized enzymes to perform this task of nucleic acid polymerization. They're called polymerases. DNA polymerases are responsible for replication, duplicating the genome prior to cell division. And RNA polymerases are responsible for transcription, the synthesis of RNA from a DNA template. Coming back to this picture we've already looked at, I want to highlight again that the paired chains are held together by base pair hydrogen bonding in an anti-parallel orientation to each other. As you can see in this picture, the strand on the left goes up from the 5' to 3' direction, and the strand on the right goes down from 5' to 3'. Because they're anti-parallel and they're held together by complementary base pair interactions, we refer to this relationship as reverse complementary. we'll say things like the strand on the right is the reverse complement of the strand on the left, and vice versa. During replication, the enzyme DNA polymerase uses a single strand of DNA as a template and synthesizes the reverse complement onto this. The starting strand acts as a template for the new strand, and so this is the term we use to refer to it. We call it the template, or the template strand. Let's see how that works here. First, we have to separate the two strands. We'll talk about this later in the lecture, and then more on Thursday. After that, the first step is a temporary starting piece made out of RNA, called an RNA primer. This is important because the DNA polymerase can only add nucleotides to existing strands. It has to start somewhere. You can think of this molecule, this RNA primer, as a temporary placeholder that allows DNA synthesis to begin. After the RNA primer is down, now DNA polymerase can come in. It recognizes the end of the RNA primer molecule, and now it's off to the races, adding new complementary nucleotides at each position along the template. After this is done, there's one last piece of business to take care of. Another DNA polymerase comes in and cleans up by removing the RNA primer and replacing it with DNA. We'll go over more details about all of this on Thursday, especially this complicated last step with the RNA primer, but I wanted to start out with a broad overview first. Before we move on from this slide, I want to come back to the directionality of DNA polymerases. They always synthesize DNA by adding new nucleotides in the 5' to 3' direction. What I mean by this is that when you look at the nucleic acid chain, you'll see the 3' hydroxyl group at the end of it. This is where new nucleotides are added by DNA polymerase. Remember that we always have the 5' to 3' nucleotide linkage by a phosphodiester bond. And each time we add a new nucleotide, we now have a new 3' end that can serve as the linker for the next nucleotide. I don't want you to memorize all of these structures, but I do want you to be very clear on the concept of 5' and 3' ends for nucleic acids. And I want you to remember that DNA polymerases always synthesize in the 5' to 3' direction. While we're talking about this, I might as well mention one more thing that's important to remember. When we write out DNA sequences like A, T, C, G, C, C, G, T, A, etc., we always write them out in the 5' to 3' direction, just like we always write out protein sequences in the N-terminal to C-terminal direction. Alright, so let's do a quick exercise to test your knowledge of this. Think back to the PowerPoint animation I just showed on the slide about DNA replication, and use what we know about the direction of DNA synthesis to tell me where the 5' and 3' ends are in the picture above. You should identify two ends that are 5' and the two ends that are 3'. If you don't remember the directionality of synthesis from the animation, you can go back and watch the video again. I'll pause here for a second to let you work on this. Alright, welcome back. So given that we know DNA is always synthesized in the 5' to 3' direction, and that double-stranded DNA is anti-parallel, we can conclude that the top strand is 5' to 3' from left to right, and the bottom strand is 5' to 3' from right to left. Understanding these concepts of directionality and complementarity is a really important foundation for all the work we're going to do on nucleic acids in this class. So if this isn't completely clear to you at this point, I strongly recommend talking about this more during group discussion, at office hours, or on the Piazza online forum. Now we're going to start going into more detail about all the steps of DNA replication. This will involve the enzyme DNA polymerase that we just saw in the previous slide, but we'll need many other accessory proteins as well. To frame this discussion, I'm going to start out by identifying 3 problems that DNA polymerase can't solve by itself. First, DNA polymerases aren't able to melt/separate duplex DNA strands to expose their bases. Second, DNA polymerases can only elongate a pre-existing DNA strand. They can't start DNA synthesis from scratch. And third, the two strands of DNA are anti-parallel, but DNA polymerase can only synthesize in the 5' to 3' direction. As we go through the mechanisms of DNA replication in more detail today and on Thursday, we'll cover how each of these problems are solved by the cell with the help of additional accessory proteins. The first problem is that DNA polymerase can't separate DNA strands. So how's this problem solved in the cell? This has to happen before DNA replication can start. If the DNA strands remain together in a double helix, we'll never be able to replicate them. Let's start out by saying there are specialized proteins in the cell that can accomplish this task of separating the strands. This always starts at a special location on the DNA called the replication origin. Replication origins are stretches of DNA that contain consensus sequences that are recognized and bound to by origin-binding proteins. We'll talk more about consensus sequences on the next slide, but for now, let's just say that these origins of replication will typically be enriched for A and T nucleotides. Remember that AT base pairs only have 2 hydrogen bonds, so this is weaker than the GC base pairs, which have 3 hydrogen bonds. Therefore, AT-rich sequences will be easier to pull apart and separate to begin DNA replication. Let's talk more about consensus sequences now. We've already covered protein sequence alignments in the class. That was during our discussion of the different hemoglobin homologs and isoforms. The same concept of lining up sequences applies here as well, but now we're looking at nucleotide sequences instead of protein sequences. The idea is the same though. We can identify highly conserved regions of nucleotides that are likely to have evolutionary conserved functional importance. Highlighting these regions of conservation helps us to identify the shared consensus sequence between them, shown here at the bottom. So like I said, origins of replication are bound by specific proteins, and this initiates DNA replication. Throughout this lecture and on Thursday, I'm going to present how this works in prokaryotes like bacteria, and then also how it works in eukaryotes like us. On the left, we have the circular chromosome of a prokaryote. Prokaryotes will usually have a single replication origin. You can see how the DNA opens up at this point, and then DNA replication on both strands goes all the way around in a circle, leading to 2 new double-stranded circular chromosomes. There are some new terms introduced on this slide, so I want to highlight these here. The first one is replication fork. This is the name we give to the DNA as it's splitting and opening up on each side of the replication origin. It looks like a Y shape or a fork in the road on each side. Really, this is the fork of double-stranded DNA splitting up into 2 single strands that are being replicated. So this is why we call it a fork. The next terms I want to introduce are parent and daughter strands. Parent strand is another way of saying template strand. This is the original strand that acts as a template for replication. Daughter strand is the newly synthesized strand. On this slide, all the template parent strands are colored dark blue, and the newly synthesized daughter strands are colored light blue. At the bottom, you can see that each replicated chromosome contains 1 parent strand and 1 newly synthesized daughter strand. One more definition to go over here is the replication bubble. These are the things that look like bubbles and start at the origin of replication and keep growing larger as the replication forks move further and further away. One last thing to mention on this slide is that prokaryotic origins of replication have a well-defined consensus sequence, but eukaryotes don't have this. We definitely have specific points on the chromosome that act as replication origins, but there isn't a simple conserved sequence that we can identify to define and predict these points. There's likely some kind of larger, more complicated set of rules governing the location of eukaryotic replication origins, but to my knowledge, these rules haven't yet been discovered. Alright, so zooming in now, let's bring some more molecular players into the game. First, we have the initiator protein. This recognizes and binds to the origin of replication DNA sequence and may act to separate the strands. After the initiator protein binds, we have the recruitment of more proteins that act as the machinery for DNA replication. I'll quickly go through them here, and then we'll cover them in more detail on Thursday. SSB stands for single-stranded DNA binding protein. These bind to single-stranded DNA and prevent it from re-annealing. Next, we have helicase. This is an enzyme that specializes in unwinding DNA. And finally, we have DNA gyrase, a topoisomerase class enzyme that specializes in relieving DNA tension and supercoiling. We'll talk about all of these in more detail on upcoming slides. First, let's zoom in now on the initiator proteins. Like I said, we're going to go over the prokaryotic version and the eukaryotic version for all of the proteins involved in DNA replication. So let's start here. The prokaryotic initiator protein is named DNA-A, and this has the ability to separate DNA strands all by itself. On the other hand, the eukaryotic initiator protein, ORC, which is really a complex of different ORC subunits, doesn't have the ability to separate DNA strands. Instead, we eukaryotes will have to rely on helicase enzymes to kick off this process of DNA strand separation. One quick point I want to make here about these figures is that all the cylinders you see here are just another simpler way to represent alpha helices instead of the cartoon ribbons that we talked about before. Usually you'll see this type of cylinder representation in really zoomed out views like this, because it makes it easier to grasp the overall structure of the protein, rather than looking at a bunch of coiled ribbons all together. Alright, that's all we have time for in the main lecture today, but we'll come back and finish up DNA replication on Thursday. To finish up the lecture now, I want to come back to the HIV virus and talk about exactly how it's able to reverse the central dogma. Let's start by comparing HIV to other viruses. HIV is a retrovirus, and we'll talk more about the retro aspect of this in a moment. But one thing I want to point out here is how closely related it is to coronaviruses like SARS-CoV-2. For our discussion today, the most important characteristic to focus on in this slide is that HIV is an RNA virus. The fact that its genome is single-stranded RNA will also come into play a little bit. It's like the coronavirus family in all of these aspects, but its life cycle and mechanism for replication are very different, so let's go over that now. For comparison, I'll show the SARS-CoV-2 life cycle first. We've already covered this, so I'll go over it quickly. First, we have receptor-mediated endocytosis facilitated by the spike protein ACE2 receptor interaction. And then, membrane fusion in the endosome deposits the RNA genome into the cytosol. After this, the viral genome is processed, replicated, and then transcribed into shorter protein-coding RNAs. Synthesis of virus structural proteins that make up the virion happens in the rough ER, and these proteins come together for virion assembly and genome packaging before the new virion is secreted from the cell by exocytosis. That's the SARS-CoV-2 life cycle. Let's compare this now to HIV. It starts out the same, with receptor-mediated fusion and depositing the RNA genome into the cytosol. But then, something very different happens. In addition to the RNA genome, the HIV virion also carries an enzyme called reverse transcriptase. And once this gets into the cytosol, it uses the RNA genome of HIV as a template to synthesize a reverse complement DNA strand. After that, another enzyme from the virion, called RNase H, degrades the original RNA template, and reverse transcriptase then comes back around to synthesize the reverse complement of the reverse complement, which is of course just the same as the reverse complement of the reverse complement is just the same as the template. And now we have a double-stranded DNA version of the HIV genome inside the host cell. The next thing that happens is also pretty new and exciting. Another viral protein called HIV integrase takes this double-stranded DNA viral genome and inserts the whole thing into one of the host chromosomes in the nucleus. This is the main reason why HIV is so hard to cure, because it gets into our genome and then it stays there, sometimes even in a dormant state. But eventually it becomes triggered to start ramping up the production of new RNA genomes and viral proteins that do the packaging and virion assembly, and that's the life cycle of HIV. The thing that's so amazing about this, in my opinion, is that the central dogma was practically considered like a fundamental law of life on Earth, and it had been treated like this ever since these two on the right pieced it together in the 1950s. So when retroviruses were first discovered, people didn't know they were retroviruses, but when they first started to study them, it took a real leap of imagination to figure out and identify what the reverse transcriptase enzyme was doing. In research, the answers we get are always guided by the experiments we design. It's easy to design your experiments to stay within the parameters of what everyone accepts as proven and possible. And in most cases, this will give you an efficient path to the answer you're looking for. But I like to think of this example of retroviruses as a reminder that in order to make really big, groundbreaking discoveries, it's important to keep your eyes open to all the possibilities and be skeptical of every assumption. That's how we discovered that the central dogma wasn't set in stone and could be turned backwards by some viruses. I don't have time to tell the full story about this, but I do want to quickly say a couple things that came out of this discovery. One is that retroviruses like HIV have been modified and adapted as biotechnology research tools for genetic engineering. And these have become such a basic part of biomedical research that they've been fundamental in the discovery of many different disease processes and treatments that have improved human health. The second thing I want to say is that the identification and characterization of the HIV enzyme reverse transcriptase was a very important step in the development of treatments for HIV/AIDS. This enzyme, along with other enzymes like HIV protease and HIV integrase, were the targets for drug development, just like we talked about last week with the SARS-CoV-2 protease inhibitors. The reason that HIV infection has become a chronic, manageable illness rather than a death sentence is because we have good, clinically tested inhibitors for reverse transcriptase and these other enzymes. The current standard of care for HIV infection is to take a reverse transcriptase inhibitor in combination with protease and integrase inhibitors. The reason to take all three at once is due to the fact that HIV is rapidly mutating and prone to drug resistance. We'll talk more about the clinical importance of mutation rates as the human disease example for our next lecture on Thursday, when we wrap up this section on DNA replication. If you think DNA replication is neat and want to learn more about research in this area, I recommend that you look up and try to talk with Joyce Hamlin, Anindya Dutta, and Mashar Adli here at UVA. If you want to know more about retroviruses and HIV, then I'll direct you to the work of Mary-Louise Hammarskjöld, Barbie Ganser-Parnios, and David Rekosh. Okay, that's it for today, but I'll see you all again on Thursday to wrap up this section on DNA replication. Thanks for watching, everyone, and I'll see you all in class.